turn it on. You have to turn it on. Nice.
Now, can you hear me? Because I do not speak very loudly. So today I'm going to talk to you about basically the overall potential of the field that I work in, which is known as synthetic biology. And before I start, I want to know how many of you have heard that term before in this room. I was hoping quite a few, because that means that the word is getting out there about what we're doing. Okay. For those of you who haven't heard the term synthetic biology, it might be easier to think about it as engineering biology. It's actually a bit of a twist on the traditional engineering of biology. And because of that twist, it's emerging as a new technology, in a way, by a number of sources. I show one here is the World Economic Forum top 10. So you see synthetic biology is on the list. Top 10 technologies is number 2, but highlighted in red are all of these other emerging technologies where, in fact, engineering biology is going to play a fairly significant role. So things like increasing the amount of food or biomass, or doing personalized medicine approaches. All of these require an understanding and engineering of biology. Okay. So I'll talk in a little bit about some of the things that we can do, but first I want to talk about why there's been a switch in terminology in my field to synthetic biology from the traditional bioengineering or engineering biology. So that still exists, there's still a lot we can do, but synthetic biology is sort of distinct, and one of the reasons for that is that in the past couple of decades, we've seen tremendous growth in our ability to read, in other words, sequence our DNA and the DNA of all the organisms that we can get our hands on, and also to write those sequences. So this chart here, that's not going to work. Okay, so the blue line in the chart on the left shows the productivity in terms of how many transistors you can fit on a chip. So that's representing Moore's Law. And most people, I think, are familiar with Moore's Law and how quickly we can increase the productivity in terms of fitting transistors on chips. And then in red is our ability to read DNA. So it's put in terms of, I think, how many base pairs, so like how many of the bits of DNA that you can read. And you'll notice that it followed for many years the same trend as Moore's Law. And then all of a sudden in the mid-2000s, it's started going much, much more quickly than Moore's Law. So we are all of a sudden capable of outpacing Moore's Law, which was sort of this, I don't know, almost godlike law in this area, and we're doing it faster. And that was because of all these new technologies that came out. Even without that, our ability to string new DNA sequences together, literally build DNA out from scratch rather than using organisms to do it, has been increasing also in terms of productivity, on pace with or just ahead of Moore's Law. So we're now able to read and write DNA as quickly as we can figure out what to do with that information. Okay, so the next big transition is that we figured out how we can recode the DNA to at least create new functions in organisms. And you can argue, Craig Venter has made a lot of waves in the past couple of years for his claim to create to have created synthetic life by basically recreating an entire genome and putting it into a bacteria and actually having that work. But he didn't really alter the functions in a lot of it, he just strung the DNA together. So what I'm talking about here is something a little bit different, where we can actually figure out what certain pieces of that information encoded in the DNA is responsible for doing, and tweak it, and still have it be functional, which is actually incredible. Because most of the time when you tweak something that's pretty optimized, you aren't going to make it any better or more optimal for your purposes. Think of, like, I don't know, a very highly engineered race car. If you took out some part of that engine and tried to replace it with a different part without fully understanding that part that you took out to begin with, you're probably not going to optimize it. So maybe if you were a special race car engineer, you could do it. But the average person on the street going in to do it isn't going to be as successful. And your odds drop even further, even if you are an expert, if you want that race car to operate in the desert on sand, right? So you might make a change, it may or may not make that car any better at racing on sand. So it's a pretty challenging problem, but we're beginning to understand how we can change little bits of the sequence and actually change the function of organisms. And so I call that recoding. You can call it any number of terms. And so we're sort of in the infancy of understanding how we can do this. So what we've been able to do, this is probably one of the more interesting slides in my talk because it outlines some of the things we've been able to do just in the past couple decades with this idea of synthetic biology, where we're really using everything that we're learning from the DNA bits that are encoding our entire life form and changing it around. So up on the left is representing all the different sorts of medicines that we used to have to rely on, say, a rare plant that only grows in a certain climate to produce a small amount of. Now we can use bacteria or yeast, the same things that are making your bread. We can have them instead produce this medicine at a much cheaper cost. We can potentially make fuels. People go back and forth about whether this is cost or energy effective in the end, but it showcases that we can do it anyway. So instead of relying on the bacteria that are in the ground, say in the soil, converting all of the decaying plant life into a fuel eventually that after many thousands of years turns into petroleum, we can speed that process up and do it in a chemical reactor inside a factory. Right? So we can do it. Whether we can do it more efficiently than the things on Earth already do it is maybe up for debate, but it showcases the possibilities. And then we can also use organisms that we are engineering to make things like fibers and fabric precursors. And so actually some companies have even commercialized processes where they use bacteria to make the main chemical that then polymerizes into your carpet fiber. So likewise, you can make the denim for your jacket using bacteria instead of plants. So kind of cool. And I think in retrospect, I work more on the medicine and fuels and chemical side of things, but after going to some recent conferences where we had a mixture of sort of art and science types, I found out where I should be is in the fashion industry, because I can make a whole lot more money making one jacket than anything in the pharmaceutical industry, which was shocking to me, but it turns out to be true. Okay. So I told you about how we can read and write DNA sequences a lot better now, that we're learning at least how we can recode organisms, and that we can use those organisms at least to synthesize some pretty cool chemicals or materials from renewable resources, right? So we can do this in a more renewable way instead of taking petroleum out of the ground. Where are we going? That's the really big question right now. What do we do next? What's the biggest possible life-changing technology that we can really develop with this field? And I'm certain that synthetic biologists will continue to develop really novel fuels and chemicals. I am pretty confident that we're going to find new ways to interface organic and inorganic systems, so living and non-living systems. So imagine bacteria that respond to a computer input via wire or even wireless signals. I think we're going to be able to do that, not as near term as making more chemicals, but certainly in the future. A lot of people are working on these things. Biomaterials is right up there with chemicals. We're going to be pretty good at that. Some of the more far out ideas are things like altering our habitat. So think of maybe encoding in the DNA for a tree. This is a favorite example, I think, of Drew Endy, who's actually at Stanford, and having it grow into the shape of the house. So we don't have to grow it and cut it down, you just have it alive and forming a shelter, right? So thinking big. Or a Kickstarter that got a lot of publicity, which is instead of having lampposts that are driven by electricity in any form, have them be trees that have the code to make a particular phosphorescent chemical that is in fireflies and have the leaves glow at night, right? So that would be cool. It got funded. I don't know that that would work because that chemical isn't really that bright, even though it lights up for fireflies. I don't know that would be enough light to walk under, but it's still an idea and one of many to come. So I think altered habitat is sort of a crazy area where anything can happen, and probably we're only at the tip of the iceberg for what can be done. And then personalized medicine is already something that's been started and will continue, right? You can, now if you have breast cancer, they will do the sequence of those cells to see which particular mutation you have, because they know that certain treatments work much better on certain types of cancer, and that depends on that DNA sequence. But that's one example. You can imagine in the future, maybe they'll look at your entire genome and see some particular gene that's sort of correlated with heart disease, but if you eat just the right foods, that won't happen. And so personalized medicine is really going to extend to the entire, making all of our lives healthier and longer. And I do think that will happen. So all of those bullet points in the top, I think, are definitely possible based on what we've seen. What is probably going to be the biggest game changer that comes out of synthetic biology is something that I can't even think of right now. And that's what's really, really exciting about this field. So anybody in the audience right now could, after seeing my talk, maybe decide to go and learn a little bit more about how to engineer biology, do the lab work, and have a brilliant idea that ends up revolutionizing the world. And so that's why I do this job, is to meet people that I can train to change the world. Okay. The problem, the reason that not everybody's jumping at this opportunity right now, is that there are a lot of challenges. And in the interest of time, I'm pretty sure I've already used most of mine. I'm only going to talk about one challenge, but it's a really big one, and that is complexity. Organisms are really complex. Nature uses evolution that is able to respond to changes in the environment very easily and survive, right? Jurassic Park, life always finds a way. When we try to engineer it, we tend to engineer pretty simple things relative to that scale, and they break easily, as I was saying earlier. So finding a way to deal with how to engineer complexity on purpose, but with a purpose, to respond to changes in the environment, and also to respond to changes that always are happening in the cell. The cell is constantly making mistakes. So if you're using a living organism to do something, it's making copies of itself. As we grow, we are having all of our cells divide. Mistakes happen. And so you have to have many, many redundancies in your system if you want anything that you engineer to be maintained. So one aspect of the major challenge of complexity. Another is that in most natural organisms, in fact all of them that I can think of, there's a lot of organization. So when you were in high school biology, perhaps you learned about eukaryotic cells, and they have a nucleus, an endoplasmic reticulum, and a mitochondria, and all of those different things. You may not remember what they do, but they all have specialized functions for the cell, right? When we engineer things now, we tend, although not in the past few years, but in the past, we sort of threw pieces of DNA in and hoped for the best. We hoped that it would just work, but we didn't really apply any organizational strategy to it. So we're going to need to learn how to do that. How do we organize things in these cells? How can we make use of what nature's done and build on that to help the cells do what we want? And beyond that, we're going to have to start working with communities of cells. And this is something I just like to throw in, actually, but it kind of fits in this area. David Nelson, who I think is at Harvard, if I'm right, East Coast, I get mixed up all the time, they took the same strains of E. coli. So imagine taking two identical bacteria, completely identical, came from the same mother cell and divided E. coli in half. And you put a green glowing protein in one and a red glowing protein in the other. And then they continued to divide and mix them equally to start. But they formed this pattern. They actually formed fractal patterns, different patterns every time they do it. And he did the same thing. He saw the same patterns in yeast. These are single-celled organisms. These aren't tissue-forming organisms like our cells are. And yet there's this pattern that spontaneously emerges in these single cells. So certainly, nature has already found a way to sort of organize communities of cells. And we don't even know what that is. We just know that it must be doing it. And so we really have to figure out how that's happening if we have any hope of engineering these to do what we want to do without any consequences. Okay. And then finally, in the last couple of minutes, I want to talk about something that is also in the category of complexity and dealing with it, and that is controlling what's moving in and out of the cells. Almost all of the applications I talked about earlier that I can predict require understanding how to get materials and information into and out of the cells. And yet that's something that we really don't understand very well. And I'm actually working on all the challenges. This is just the one that I'm going to highlight here with a couple of examples. Oh, I should back up for people that have not seen a cell in a while, which is probably a lot of you. Okay, so this is a cartoon of a gram-negative bacteria. So think E. coli, some bacteria that is typically a pathogen in our gut, because that's what we've tended to study in the lab. And you see the DNA is all coiled up in the center. You have this blue stuff, that's called the cytoplasm. That's sort of the soup where everything happens for a bacteria. You have these long, stringy things, those are the flagella. And the dividing lines, very thin lines, those are the membranes. That's what's actually controlling what's going in and out of the cell. And I've blown them up to show they're basically lipid bilayers if you are interested in the chemistry behind them. And so that separates the cytoplasm from the stuff outside of the cell. And there's a little tiny part called the periplasm in this particular example, but it's not that important to what I'm trying to convey. So this membrane is amazing, really amazing. So at the macro scale, in our world, we've developed a lot of different membranes. I'm sure you've heard the term membrane, and it's been applied to many things. And this membrane for this little bacteria that's existed for many, many thousands of years has the ability to control the flow of electrons, ions, so something like a reverse osmosis water filter, gases, that's a contact lens, which actually has to have very precise gas permeability to get the right nutrients to your eyes, and all sorts of other compounds, very specific compounds, depending on the organism. So bacteria maybe to let sugar in and to spit out antibiotics, right? So all these things are controlled by the same membrane, and it also responds to changes in temperature. So it's amazing. We have all sorts of different materials to do all of this, and this little membrane does it. Well, we need to figure out how, because we need to control the movement of chemicals. We need to be able to control what sugars go into the cell and what products come out. We need to be able to control electron flow if we want to make mimics of photosynthesis. That's a great way to convert sunlight to energy, much like the earlier speaker was talking about. But if we can't control electron flow across that membrane, that's not going to happen. We're not going to be able to use biology to do that. And many other applications. One that I put on here is biosensing. So detecting, say, toxins in the environment. If you want to spray over the desert a bunch of bacteria and look to see which ones are glowing because there's some chemical or explosive nearby, you're going to be able to transmit information across this membrane to tell the cell to turn green when it senses this chemical, right? So all sorts of things that we need to control the movement across that membrane. And that cartoon I showed earlier was missing a really key component. So that was the traditional cartoon that's in a lot of textbooks. And yet actually that membrane is filled with these globules called proteins. So similar to the protein that you have in your diet, but actually all of your cells have protein. It's sort of the worker bees of the cell that do all the functions. And many, many different kinds of proteins sit in the membrane. And you can ignore all the writing on the cell. I just wanted to have the picture, and I didn't cut it off properly. And so we need to figure out which proteins are controlling which actions, basically, the flow of which materials across the cell, and see if we can make use of that. And the way I see it, I come from a discipline, actually, before I was a synthetic biologist, where we were figuring out how to manipulate individual proteins to do new functions, slightly different functions. So there's a protein in your body called an antibody that binds to all the different potential viruses or toxins that are in your blood and clears it out for you so you don't get sick, right? So we figure out how to make a small change to that antibody and make it better. And so that's called protein engineering, that idea of just making a small change to these proteins to do what we want it to do instead of what it naturally could. And we can use that same sort of technique to change these proteins that are sitting in the membrane and change what's going across the membrane, what materials are going in and out of the membrane. But there's really only two things we can do. We can change which molecule it acts on, or we can change how many of them there are. So we have two knobs to turn, and so that's what all of the people that work with me at Berkeley are doing right now. And so I'll just give one and maybe two examples depending on that time, so we'll see. So one example of what we did is we took one of these, it's actually many proteins, not just a single protein, so there's 9 in this particular structure that sits in the membrane of E. coli. And it normally functions to pump out antibiotics. So antibiotic-resistant strains, one of the ways they're resistant is they actually just pump it right back out. They have these pumps in the membrane that secrete it out of the cell, so it's not going to hurt the cell if it's not inside anymore. And so we took that pump, we made actually just 2 changes in the DNA. So we changed 2 different bases of DNA, you know what the bases are. And we made a pump that was almost identical, only now, in addition to acting on the antibiotics, it also was able to pump out butanol, a potential biofuel. And that increased our yields of butanol, when people try to make butanol in cells, by 40 to 100%, depending on which way we did it and which mutation we're talking about. So we can use the strategy of nature to help get out some of these chemicals that we're tracking. And I included this picture on the left. This is the cover is actually done by my brother-in-law, who's an artist and was inspired. So this is his rendition of a butanol molecule kind of punching out of the cell. those of you who are wondering about that image. It's a little different from most science talks if you've been to many. Okay, and one more thing that we've done is, on the other hand, we want to make materials like spider silk. You can't make a lot of spider silk, but it's a really cool material. Because when you try to make a lot of spider silk, you need a lot of spiders. The spiders are very territorial, so you don't get a lot because they kill each other if they're too close. Okay, so one of the ways we can make spider silk is actually by taking that DNA out of the spider and putting it in bacteria so that the bacteria make it. Unfortunately, silk polymerizes into fibers, so you don't want that to happen inside the bacteria because it kills them. And so it has to go out of the bacteria. And we figured out all of this. We figured out how to get bacteria to make it and secrete it out using one of the particular proteins in the membrane. But it didn't do it very well. So we engineered it to have many, many more of these proteins in the membrane. So that other handle that I said, we could increase how many of these transporters are there. We just took one that worked and made about 10 to 100-fold more. And so up at the very top on the right, I have what's called a Western blot, and all those black things that's how much protein that we have coming out. So the darker the black spot, the more protein we got out of the cell. And so you can see as we went through different trials of engineering the cell to make more of these, we got more and more protein out. And eventually we got enough that we could actually make polymers out of materials that we were secreting, and we even were able to engineer those polymers a little bit. So we made things like a polymer that has an antimicrobial peptide. And so that particular material is the one on the right on the bottom. So if the cells are alive on it, they're red. If they're dead, if they're dead, they're red. And if they're alive, they're green. And we found that when we used that material instead of the previous generation that didn't have that antimicrobial feature, that all of the salmonella that we tried to embed in the material died, almost. You can see a few green spots in there, but most of them are dead. So this is something that could potentially be used to coat implants, right? So you don't get infections in your implants. So the potential for making materials is just enormous, and one of the ways we can do it is by making that bacteria. So that is all I really have to say. This is my last slide, and it summarizes that we just have enormous potential from engineering biology, but there are a lot of challenges to overcome, some of which I've touched on here, dealing with the complexity. But I think if we do that, we can do almost anything we really dream of with biology. So with that, I'll thank my group, all the people that worked on many projects, not just those I've shown, and some questions.